Awesome. Thanks, Allison, and thank you everyone for bearing with us in those for those technical difficulties. So welcome to PGTME's 5th Annual Lunchtime Lecture Series. Like Allison said, my name is Aiden Ilmus. I am the program coordinator for the working group on pediatric gene therapy and medical ethics. And our speakers who are joining us will introduce themselves shortly. We're so grateful to have you all here for our 5th year of this event. We are always impressed and inspired by the diverse group of attendees and thoughtful engagement. PGTME is a project of the Division of Medical Ethics housed in NYU Grossman School of Medicine's Department of Population Health and is funded in part by a gift from Parent Project Muscular Dystrophy. You can learn more about PGTME on our website. John will drop a link to that in the chat, and you can join our mailing list to receive our quarterly newsletter to keep up with our work. We're very excited to have you here today and to hear from today's speakers for this session called The ABCs of Genetic Medicine, What's in AAV Anyways? If you are unable to attend any of our talks today or for the rest of the week, we'll be posting captioned recordings once the week has concluded, and we'll share that link with all registries. Today's event will feature brief introductions by our speakers, followed by a presentation and a moderated discussion. When you registered for today's session, you were asked to vote on which gene therapy related terms were of the greatest interest to you. Our speakers will be explaining the highest voted topics today. We've also allowed at least 15 minutes for audience Q&A at the end, and we encourage you to submit questions or upvote other participants' questions through the Q&A feature throughout the panel. And with that, I'm going to hand it off to our speakers today to introduce themselves and their work and get started. Tim, I'll pass it off to you to begin.
Yeah, hi, I'm Tim Cripe. I'm the Division Chief of Pediatric Hematology, Oncology, Blood and Marrow Transplantation at Nationwide Children's Hospital. Been privileged to be here 12 years and watch Kat Meyer and Jerry Mendel and Kevin Flanagan and Brian Kaspar and many others develop really cool AAV gene therapies, and to learn all about that as I go along on my cancer research journey, and also now developed our own AAV platform and spun off a company called Vironex Therapeutics, developing AAV gene therapy for cancer. So also, I had the privilege of giving a lecture in a graduate course that Kat organized or co-organized at Ohio State University for graduate students, mainly on gene therapies, but a lot on AAV. So with that introduction, I'm going to serve today as more of a sidekick. I think of it like the NFL, play callers. One gives you the facts and the other adds color commentary. So I'm going to be that analyst adding the color commentary to Kat's presentation, and also I'll be keeping a close eye on the Q&A list and try to bring forward any questions that arise. So Kat, turning it over to you.
Yeah, thank you, Tim. It's a real pleasure to be on this panel with you today. So my background is cellular and molecular biology, but I moved to Columbus, Ohio 15 years ago, really because I wanted to work on gene therapy, and I knew it was going to happen in Columbus, Ohio. So I joined Brian Kaspar's lab back then, and we were able to translate the gene therapy for SMA to the clinic, which is now one of the few AAV gene therapies that is FDA approved. It's called Zolgensma. Many of you might have heard of it. Of course, now there are others as well. And in the last 15 years, actually, I was then able, based on this initial experience, translate another 5 gene therapies to the clinic, which are in different stages of clinical trials. I work with a lot of patient foundations as well, and I've seen a lot of these programs move forward through the translational pieces. In May this year, I joined Alcyone Therapeutics full-time as their chief scientific officer. I still have good relationships, of course, with Nationwide, but I'm testing out the industry side at this point. So let me share my screen. And this is a mix of different presentations that come together based on what you guys wanted to hear about, based on your voting. So I'm sorry that the layouts are a little bit all over the place. So, but I think you should be able to. Hopefully this will be helpful, and you should be able to follow it, even if the layout is a little bit different. I'm trying to get this into presentation mode. Now, hold on, 1 second. All right, does that work?
I think you need to swap displays because we're seeing there you go.
Still?
Perfect.
Good.
Yep.
Okay. Good. Okay. So, and I really want to kind of start from scratch here a little bit with what is gene therapy, because you hear it all the time, but it is not actually perfectly obvious. Technically, as a definition, gene therapy is just a transfer of genetic material to either treat or prevent diseases. And there are different types of gene therapies. Gene replacements or gene addition, gene silencing, or gene correction. Gene replacement, which you might hear most often, is actually a terrible term, in my opinion, because oftentimes it doesn't refer to replacing anything. It just means adding a new copy of the functional protein, and I'll illustrate that on the next slide as well. But yeah, so that's something important to keep in mind when you hear the term gene replacement. In most cases, what is meant is actually a gene addition. And then gene silencing is a technique in which we are trying to reduce the expression or abundance of a aberrant toxic protein or RNA. And lastly, gene correction is really the most precise version of gene therapy, where you go in and you try to correct specific mutations or specific areas of the patient's genome. And in most cases, this is done outside of the body, oftentimes for blood cells, which you can remove, you can fix them, you can verify that they're doing okay, and then you put them back into the patient. Although recently, there are new technologies that allow to do this also in vivo in the patient. And that's going to be, I think, an exciting development as well. To date, gene therapy is mostly used for monogenetic diseases, which means we kind of need to know what's the underlying cause, what's the mutation, in which gene is it, so that we can correct it. But there are also approaches that are trying to generally target common pathways of diseases, and we'll see how that moves forward in the clinic as well. And please, Tim, you'll stop me if there are questions that need to be addressed.
Actually, so I'm gonna type some answers, but one question has already come in, has been, would you consider genetic material to include RNA or DNA only? This can be confusing. You want to answer that live?
Whether it's confusing, RNA or DNA only. So any genetic material. So RNA therapeutics so the definition can be used in different ways, but technically RNA transfer is genetic material as well. RNA, as many of you know, viruses, for example, do contain, many viruses have RNA genomes even. So this is certainly a type of gene therapy in the broader definition. I hope if that answers your question as well.
Yeah, I think I would just add that, for example, the COVID vaccine is an RNA insertion into cells to produce the COVID virus protein that created the immune response. But RNA tends to be short-lived, and that's an example of a short-lived gene transfer. And so I think it's in a different category than gene therapy, where you're looking to really modify the genetic expression of a cell long term.
Yeah, I think that is probably a good point, Tim, where the confusion comes from, because technically gene therapy, in the mind of people, it's always long lasting, right? But it's true that, for example, antisense oligonucleotides or RNA therapeutics don't have to be long lasting, but they're still in this category of gene therapies. So yeah, that's a good point to make. But in most cases, if somebody talks about, I'm going to develop a gene therapy, they think about a long-lasting gene therapy, which in this case is usually DNA-based.
Yeah, DNA definitely lasts a long time. Usually it goes into the nucleus and stays there until that particular cell dies or divides, and then sort of spreads that gene amongst its daughter cells and gets diluted out. So I think most of the time it's DNA.
Yeah. So this is kind of just in very simple terms saying what we're trying to explain here. So a cell that misses a functional protein or functional gene, we can come in with a gene therapy and basically fix that cell so that the cell is able to make the protein itself again. And I think that's something that fascinated me from the beginning. So you're giving the cell its own treatment, and the cell will keep producing that treatment over time. So there are many types of gene therapy. And again, it comes back to like, are we talking about RNA or DNA? Are we talking about something long-lasting or non-long-lasting? So I'm just going to list them here for you. Of course, there are viral vectors. And for the viral vectors, you can have integrating viral vectors, which basically take that additional gene and put it into the patient's genome. Or you can have non-integrating vectors, which basically just persist to a certain extent or can be very long-lasting independently. So they're not integrating into the patient's genome. And it's just a different types of viruses that lead to these kind of reactions. And then you have non-viral systems, which again can be integrating and non-integrating. Mostly used are the non-integrating non-viral systems. So those are nanoparticles or physical transfection of the agent. And I'm going to have a picture about the different methods on how can you get DNA into the cells in patients. Just a little bit further down. But just you can see there's a lot of options, and depending on the target tissues, some are more suited than others. It really depends on what you need to get done and how broadly you need to spread your gene therapy. You probably heard about what a virus is. Just really quickly, viruses can only replicate within a host organism. They are very simple, very small structures, and they are very diverse. As I mentioned, they can have DNA or RNA genomes. They can be single or double-stranded, but importantly, they will not replicate without the host organism.
And I'll just mention those when you say integrating, some of these viruses naturally integrate, meaning they insert a fragment or their whole genome directly in line with the DNA that's in the cell. So it becomes part of the cellular DNA. And then that's when it makes it more and more. Every time the cell divides it, it recreates it and divides that as well. Whereas if it doesn't integrate and insert itself into the cellular DNA, it's outside. We call that extra chromosomal or outside of the cellular DNA. Episomal is another word used for that. And those generally don't, sometimes they do, but sometimes they don't get reproduced as the cell divides. They just get diluted out if the cell divides. And that's really based on the biology of the virus you're using.
And, obviously that changes your choice of virus depending on which cell type you're treating. If you're treating a cell type that's very rapidly dividing, it doesn't necessarily make sense to use a virus that doesn't integrate because, as Tim said, it will dilute over time. So what is a viral gene therapy or a viral vector? So here you look at a very simple scheme of a AAV vector and an AAV virus. On top is the virus. The virus contains, so that yellow little box is the virus, and inside the virus there is the genome. The genome contains information that the virus needs to replicate, to make more of itself. And in gene therapy, what we do is we use that yellow box, the same surface of the virus, but inside we're transporting the information that we want to make, which is the therapeutic protein. So there are no viral genes left. So we have engineered this virus to transport our therapeutic gene. So it's really just kind of a mailbox, if you want to think about it that way. A tool to deliver your message, what you want to deliver to the right place. And we call those viruses, quote unquote, vectors or recombinant viruses. So that might be a term that you've heard before. Tim, did you want to add anything here?
No, that's good. No questions yet.
Okay. So that's about viral vectors. Now, non-viral gene therapy, which we're going to talk a little bit less about today, but just so you know, there's a lot of different methods on how you can deliver those DNAs or RNAs into the cells. And some of them are physical. You can laser. There are gene guns where you can, with speed, you can inject basically genes into cells and tissues. You can use certain chemicals as well for better uptake, or you can inject, for example, genes directly into an embryo or something like that. But then you also have the ability to package your DNA or RNA, which is on the bottom half of this picture, into these nanoparticles. And the nanoparticles can be any sort of chemical assembly to which your DNA or RNA or even proteins can stick to. They can be either loaded inside the particle or on the surface of the particle, and it will allow to spread your whatever you want to deliver throughout the body. And of course, again, the choice of delivery here really depends on where you want to go and how far you need to go and how big the spread needs to be. For example, it's relatively straightforward to develop a liposome that will target the liver, and you can use specific proteins on the surface that will increase the uptake in the liver as well. But if you have to bring something to the nervous system, that's a whole lot other system, or to like muscles, which are everywhere in the body. That's a lot more complicated because it's not as concentrated and located as the liver, and it doesn't have the same amount of blood flowing through as well. Okay, so this goes to what Tim just mentioned. We have viruses that integrate into the genome of the patient and become part of their endogenous gene. And there are viruses that basically just remain as episomes or almost like plasmid-like structures in the nucleus. And those tend to get lost over time if the cells divide, or can be lost over time. And I don't know how important this is here, but wild type AAVs. So AAVs is one of the viruses we use a lot, which we'll talk more about here. They exist naturally, and the naturally existing AAVs, they can integrate into the genome of the patients or healthy people. But the recombined AAVs tend to not do that, or to a lot lower extent. And that is one of the advantages of AAVs compared to other viruses. It makes them safer because there is less of a risk that they destroy something where they place themselves in the genome. So why are we using viral gene therapy? It sounds a little bit science fiction-y here. We're taking a virus and removing things and adding things. It's really, to date, the most efficient and most long-lasting way to get materials to a large number of cells throughout the body. It does also carry the highest risks, I would say, but again, given the efficiency, I think it is very tempting. In most cases, because there are risks associated with these therapies, we use them when there are no good other treatment options, or there is substantial room for improvement of the treatments. Oftentimes, these are severely debilitating disorders, so the benefit-risk ratio needs to be in favor of the patient, of course. And the really interesting part is that with a single treatment, you only have to take this therapy once, you can potentially have a life-changing, long-lasting effect, which obviously is really, really intriguing. But what we don't know, and there's a lot of things we don't know, is whether symptoms that have occurred before the therapy are reversible. As you can imagine, that also is going to depend on the type of virus that you use and on the type of disease. For example, if the disease is degenerative and the cells die, then there is no way that we can bring the function of those cells back, and the damage that has occurred might be lasting to some extent. So that's something to keep in mind as well. So what are adeno-associated viruses? It's one of those classic examples where, we make incredible discoveries by accident, I guess, or just by coincidence. The adeno-associated viruses were discovered as a contamination of adenovirus prep. So the adenoviruses are much larger viruses that we use, for example, for vaccinations and for other uses. And in those preps, they realized these smaller blurbs, which were happened to be adeno-associated viruses, which is kind of it's a virus that isn't very functional on its own. It can only work even in its healthy state in combination with other viruses. It cannot propagate on its own. So it needs adenoviruses or other viruses to do that. But for us, this is a really good tool to work with gene therapy because it has a low immunogenicity compared to some of these other viruses that we've used. The AAVs, or adeno-associated viruses, which you can imagine the name comes from having it discovered in these adenovirus preps, come in different flavors, which we call serotypes. And that just simply means that the surface structure, the composition of the surface of the virus can be slightly different. So there can be minor changes in proteins that are on the surface of the virus. And those changes will impact which cells the virus will bind and how efficiently, as you can imagine, because the surface proteins are important for the virus to attach to the cells. So if those are different, you will get a different pattern. And of course, the viruses that we know best are the ones that we've studied for the longest time. So AAV1, AAV2, AAV9, those are viruses that we've used for a long time because they were discovered earlier on. But there's a lot of effort in the AAV field to create new versions and maybe evolve the virus even artificially to get more specific cell types or only transduction in a certain organ so that we could reduce some of the side effects. I said that AAV viruses are low immunogenetic, that's true, but there are still a couple of different immune reactions that will occur when you get infected with AAVs, and that contains both cellular reactions from T cells, as well as humoral reactions, which are antibody-based, and then even some of the endogenous immune reactions, such as just toll-like receptor recognition, et cetera, et cetera. And the immune reaction is very variable between patients, and we don't really completely understand yet why some patients react stronger to the same amount of virus compared to other patients that have a much lesser immune reaction. And those are important factors to consider in the clinical trial, because a dose that is safe for some patients might not be safe or might create some issues for other patients. Tim, did you want to add anything here? I'm not sure.
I will just say immunology is extremely complex, hard for everyone to understand, including those who study it intensely. So don't feel bad if it seems mysterious, but we're happy to answer questions as we go with this. And this is a complex diagram, but I guess the point is, as you said, there's lots of different immune responses that can occur, that pre-exist, or that happen after you get the gene therapy. Usually the pre-existing levels of antibodies are much lower. Once you get a high dose of gene therapy, it goes sky high and becomes more difficult then to dose later. And those are all major issues within the field, but we can talk about those as we go along if people want to type questions in.
Yep. So yeah, so what are the challenges that are related to these gene therapies? Certainly the immune reaction is a big challenge, and there are two types of immune reactions. It can be just to the virus itself, or it can be to the transgene. You think about, when we develop these gene therapies, in some cases, the patients actually have never made that protein. So when we bring in the protein, there is a risk that the patient would recognize it as a new protein, as an intruder, and would actually attack the therapy. So that is certainly an additional risk, depending on the types of mutations that we're treating. But the most challenging reactions so far that we've seen have been to the vector, to the proteins of the virus itself. One big risk that we have to mitigate is the targeting of unwanted cell types. We obviously select a virus that we know targets the cells that we do want to target, but oftentimes you can't completely get rid of off targeting. So for example, if I want to target the nervous system, even if I put the virus into the spinal fluid or in the brain directly, it will still leak out into the blood and it will still go to the liver. So if this is a protein that the liver cells can't deal with, then that could be a problem. And then, of course, as we said, another risk can be insertion into the genome. It's very rare with AAVs. That's why AAV is one of the favorites when it comes to gene therapy in the patient, in the living patient, instead of outside in the petri dish. But it could happen that aberrant mutations occur because of the virus integrating into the genome. And then the biggest problem, I think, is, which makes the translation of these therapies difficult, is gene therapy is not reversible. So once you've done it, it cannot be altered. So if you do have a bad reaction to the transgene or to something in the vector that stays, we have to deal with it. We can't take it out. So that's one of the big challenges. It also means that we have to be very careful how we select the dose, right? If we put too much, it can be very dangerous. If we don't put enough, it might not be effective. And as Tim said, redosing at the moment is very challenging. We hope that will change over time. There is a lot of people that are working on this topic. But certainly for now, it's kind of a one and done thing. So if we miss the dose in our translation, it will be very difficult to redose.
A couple questions have come up. You said that AAV is currently the best way to quote unquote administer gene therapies, but the problems with them have been known for many years, especially immunogenicity. Are there any promising vectors on the horizon?
Yeah. Well, I think it's a good question. So yes, AAV has problems, and I actually was going to point that out right on the next slide here too. Even though we're saying it is an amazing vector because it is less immunogenic than other vectors, it has unfortunately created situations in which patients died in several clinical trials, and also with Zolgensma, which is a drug that's, I think, has done a lot of good for SMA patients. But what we're seeing really is high dose. Above a certain dose, it becomes very dangerous for the patients. Side effects that have been seen are liver toxicity and hyperimmune responses, as well as cardiac problems. And all of these are generally related to a very high dose. So I think, strategies that we can employ is ways to reduce the need for these high doses, because if you put into context, the amount of vector that we're giving the patients is more than stars in the Milky Way. It's an incredible amount of virus. It's not just like getting the flu. These kids are getting billions of viral particles, and there are ways to reduce the amount of virus needed with better delivery systems, with potentially adjuvants that could increase the uptake of the virus. And then also, as I mentioned, with searching for capsids that might be more specific for certain cell types or certain organs. So I think we're only scratching the surface. And then the second problem is we just need to better understand the immune reactions to the virus, because if we would understand why some patients react a lot more aggressively to the treatment, I think we could address that too, and then that would allow us potentially to use the doses more safely.
Another question that arose is, does the immune risk of AAV make CRISPR a better method for gene therapy? So we may need to explain a little bit about the differences there.
Yes, and I do have some slides about CRISPR because I know a lot of you have had questions. So CRISPR, or any genome editing method, is a tool that still needs delivery. So you need to get that CRISPR into the cells, right? And you could use non-viral delivery, or you could use viral delivery. The drawback always is, how many cells do you need to reach in order for the therapy to be effective? And as I mentioned to you, in most cases, depends a little bit on the organ, but in most cases, the viral delivery is more effective to date than non-viral particles. Exceptions might be the liver, which is easily accessible. But in order to get your CRISPR to the cells, you need to deliver it.
And the CRISPR can also be immunogenic in its own right. It's a bacterial protein. And
correct.
Because we have a microbiome of bacteria, some of us probably already have immunity against CRISPR. That's why I think most of CRISPR has been used ex vivo, where you take the cells out, manipulate them with CRISPR, then give them back. Although we are now in a world where there is in vivo CRISPR happening.
Yeah, I think that's a very important point, Tim. And the in vivo CRISPR approach, I think they're delivering the RNA, not the DNA. So it's not going to be a long-term expression of the protein, because as you say, and there are papers out there already that say that a good fraction of humans have antibodies against the CRISPR, the Cas9 proteins. So this is another thing that I think CRISPR-Cas is a very interesting system, but also comes with a lot of considerations to think about. You don't want to continuously express a bacterial enzyme in your body over time, especially not an enzyme that is cutting your DNA. So you have to find ways where you can, over a short amount of time, express this protein so that it can do its work and then disappear. And when you do that on a short term, it's usually less effective because you only have a short time for the protein to work. So lots of things to figure out on that end as well.
Right.
Yeah. So the problem is, and that might be a bit confusing also for people. It's like, why is this happening in the clinic? Why don't people, make sure that this is safe when you go to the clinic? Well, we do, we try, we work a lot on small animals, on mice, and also on large animals to try and bridge the ideas and the concepts to the clinic. But what you need to keep in mind is that when we work with the mice, they're inbred, so they're all genetically similar, and they have basically the same food, the same treatment every day, lights out at 6, lights on at 6 in the morning, and that's still the same with large animals, and that's not at all the case for humans. Humans are genetically very diverse, but also their lifestyle, their care, additional medications, all of that is different between each individual patient. Also, in most cases, a mouse represents one mutation, maybe not even just a knockout of the entire gene, doesn't represent the types of mutations that we see in the patients. And in large animals, because they're very expensive and difficult to work with, we usually only treat healthy animals. And healthy animals sometimes, they just do better with these kind of immune challenges compared to patients that are already, sick or have, pre-existing conditions. So there is just some things we cannot model preclinically that unfortunately we do have to use the clinical trials to establish the best doses for the treatments, etc.
Very good. Keep going.
Yep. So then one thing amongst, I think, that I find very difficult when people read these papers or they talk to each other, maybe you talk to a patient foundation, oh, we had this dose for our patients and that worked really well and it's a lot different from the dose that you want to do. You can't really compare the doses between the programs. That's very confusing and complicated because the methods that are used to determine the concentration of the virus is very different. So you have to imagine that you're looking at a bottle with water and sand in it, and you're trying to count the number of sand corns in the water. It's impossible, right? So you need to come up with a strategy how you can approximate the number of particles that you have. And there's different methods. You could, for example, measure the density, how much light goes through your bottle. It will give you a certain number. Or you could take a small amount of your liquid, count that, and then multiply. But depending on which method you use, you will get a very different answer. And it doesn't change the amount of virus that you have in your bottle, but it changes the way you label it. And so comparing doses between clinical trials is incredibly difficult, if not impossible. So you really have to, if you work with researchers, you have to really trust your own program to come up with the best translation for your gene therapy, is what I'm saying, and not look at maybe what other people used in the clinic, or not as much. So the other thing that, as I mentioned, the immune system is highly variable. But for example, we work with injections directly into the spinal fluid. There is leakage in certain diseases, and the circulation of the spinal fluid is different between patients. But even certain metabolic factors can change the uptake of the virus. So this is preliminary data from my lab back at Nationwide, where we used just an AAV with a green fluorescent protein at a low dose, and we manipulated the mice with metabolic factors, and you can see it changed the uptake of the virus between the different groups. So lots and lots and lots to figure out for us on the research side for that. Another big problem, and then I'm going to stop talking about AAV problems because I'm a huge fan. I mean, maybe it doesn't sound like it, but I am a huge fan of the technology. But when you deliver the virus, and I think that's true for any virus, not just AAV, it's not going to just evenly spread throughout the body or through your organs. It's going to be patchy. The uptake is going to be very patchy. There's going to be hot spots where you're going to have more virus, and there's going to be areas where you're going to have less virus. So it looks a little bit like the skyline of Manhattan. And that's very difficult too, because how do you ensure efficacy? So you need enough in the areas that don't get as much virus, and you shouldn't have too much in the areas that are easily targeted. And it has to do sometimes with the exposure to blood and to the spinal fluid, and deeper structures are usually more difficult to target as well. Tim, did you want to add anything here?
Oh, I think that's a great point. And it just illustrates that, we're still just in the infancy of this field, really, in terms of not only understanding, but being able to control like where it goes, et cetera. So there's still a lot of research that needs to be done. And the fact that we have some FDA approved versions of this is really amazing, but it's not the end of the story.
Yeah, very much so. And I think there is a bright future. It requires a lot of work and research. So so this is kind of, I think after the first really big successes, people, I think, forgot how complicated it is to develop these drugs and how much work went into those initial programs. And so there was a lot of excitement, and then unfortunately there were some frustrations with reported issues in the clinic, as you know. But I do think, based on the recent developments that I see, there is more enthusiasm and there is more excitement again, that we are on the right track to hopefully understand this technology better and make it safer over time. I was gonna switch gears a little bit and talk about genome editing moving forward, or unless there are any specific questions about AAVs and
there's no questions at the moment. I think we move forward because this is also an important point.
Great. Okay, so genome editing. So that's really going in, changing the DNA, changing the hard copy of a person. There are different tools. Some of them are older and more complicated to use. The ones that you probably have heard about are zinc finger nucleases, TALENs or transcription activator-like effector nucleases, or then the CRISPR-Cas system. And all of them are a little bit different. And I wasn't going to go into the details for each of them, but ultimately it has to do with how they recognize the DNA. That is one of the biggest differences. The zinc finger nucleases recognize triplets of the DNA pieces, so a little bit bigger piece, and you have to very carefully design them and find the proper form that will exactly find those triplets that you're looking for. The TALENs, they identify individual nucleotides, so that's already a little bit easier. Still a little bit complicated to design. And then the CRISPR-Cas9 system uses what we call a guide RNA, so a small strand that is actually complementary to the sequence that you want to cut. And it is very easy to design. So ultimately, the ideal properties of a genome editing tool is you want it to be very specific. You don't want to have off targets. You only want to modify the gene you want to modify, nothing else ideally. It should have a high efficiency, because if it's very specific, but it only functions, in 1 out of 100 cells that you target, that's not going to give you efficacy in the clinic. Of course, you don't want it to be toxic if at all possible. It should be easy to design, have low costs ideally, and be broadly applicable. And out of those 3 systems that I just mentioned, CRISPR-Cas really kind of hits almost all of those. It's easy to do, very easy especially to construct, and it has a relatively high success rate, good activity as well, etc., etc. Toxicity in humans, as we said, we don't really know, but there's certainly in the animal models, it has catapulted, the generation of new animal models, for example, over in research. It's been an incredible tool for that.
I'll just comment that these are all proteins, or they're all based on proteins. That you need a protein to conduct an enzymatic reaction, which is what you need to switch out nucleotides in and out of a DNA, to cut some out or to insert some in. So they're all going to be immunogenic in some way, because proteins have lots of antigens that the immune system recognizes.
Yeah, that's a very good point. So these are all going to be new to your body, right? Not something that you usually carry around. So how does this work? And I'm going to just focus on CRISPR, because this is really, I think, the tool that has been taking over the field. So the CRISPR-Cas9, or there are other numbers, 7, 8, 13, 12, whatever. It's different proteins, but the idea is the same. It's actually been discovered as a defense mechanism in bacteria. So bacteria have this mechanism how they can defend themselves against viruses, in which they basically keep memories of those viral sequences and scan for new infections. And when they find a new infections, they have this protein that's called Cas, which will cut that DNA, that attacking DNA, basically. So the guide RNA is basically searching for something unwanted in the bacteria. In our case, it will be searching for the stretch with the mutation that we want to replace, and then the protein will bind where the guide RNA indicates. So it's really like a search engine, basically. The RNA is a search engine, and then the protein binds there, and then executes this enzymatic reaction, which induces a cut in your genome. And what results from this in different ways, and I'll show you a little bit in the next slide, I think, what are the different options, is that you can really modify the sequence right there. So here are a couple of examples. The first one is to just make a cut and then rely on the cells to basically fix it. And if that happens, you can either get a deletion of that area, or you can get a random insertion of some pieces as well. So sometimes we use this to create mouse models where we are trying to destroy a gene, for example. But you can also use it to get rid of a mutation and rely on the cell's own ability to fix the problem correctly. Another way to do this is you cut the DNA and you provide a template, and that template will be used and integrated into your DNA. So that's where you would replace, for example, a mutated piece. And then some of the applications that you might be less familiar with are actually not cutting the DNA, but really modifying the activity of the genes. And these are called deactivated Cas9 or dCas9, you might have seen, which basically in this case, the protein has been rendered non-functional. And so it can still bind the DNA, but it will not cut. And by binding, it can inhibit the transcription of the gene. So you can reduce expression of a toxic gene, or you can fuse the protein with an activator sequence, And you can make more of the protein just by having the protein sit there. Now, of course, the drawback of the last option here is that in this case, you do have to express your Cas9, your bacterial protein, permanently in the cells, because the effect is only going to happen as long as the protein is there. And that, again, is associated with significant risks of immune reactions.
That's pretty complicated stuff. But we do have one question, but it's related more to the AAV system. So
Okay, yeah.
for this, we'll go back to that.
Okay. So yes, so I was gonna stop here talking about the CRISPR system. So when you see the numbers Cas 9, 7, 8, what that means often, or sometimes you see SP Cas9 or SC Cas9. So those are just, they're different proteins that have been isolated and identified from different bacteria. And some of these proteins actually don't even act on the DNA. They can modify RNA as well. Cas13, for example, I think is an RNA modifying enzyme. So in that case, they still do the same thing. They cut, but they don't cut DNA, they cut RNA, which could potentially be safer, but again, with the drawback that you would have to have the protein there long term, because the RNA keeps being made from the gene, right? Why do we have so many different versions of Cas9 or Cas systems? It's because of the delivery problem. The enzymes are actually relatively big, and it's been very hard to engineer systems that will fit, for example, into AAVs. So people had to become creative to find, the smallest versions of Cas in bacteria, or even engineer them so that they can package it into the viruses that we have. Again, why would we use viruses? Because it is the most effective delivery tool that we have to date. So there's a plethora of proteins that have been involved. Also, of course, some of the evolution happened because we wanted to be more specific, because there isn't to date any protein that will exclusively only 100% cut that one position that you want to. Unfortunately, there's always side effects that you have to think about with this kind of technology. So the efforts that have been there is to find more precise enzymes and to find enzymes that are more efficacious and smaller, so we can put them in our AAV.
So those are some challenges. There's a question, what are some of the other challenges with CRISPR therapeutics?
Yeah. So certainly the efficacy is problematic, and it depends on the cell type. So rapidly dividing cells are, based on the experience we have so far, are easier to modify than cells that are not dividing anymore. So for example, neurons, even if you target 100 cells and every cell gets your CRISPR system, only maybe 1 or 2 of them will really implement the change that you're looking for. And so it becomes a balance between, how long do you express your Cas protein so it's not toxic, so that you can maybe get more modification of the cells, versus, is there going to be a toxic reaction to the protein itself? I think, again, these are all things that over time, because a lot of people are working on it, are probably going to get more precise, more efficacious. But I think, I worry sometimes that right now there's a little bit of the misconception that we're already there. This technology is ready for broad applications in patients for any disease. And that I don't see is true, in my opinion. I don't know how you think about it, Tim. But certainly for the nervous system, I am very skeptical. I'm a little bit worried about it. There's certainly a lot of work that needs to be done.
Yeah, I think it's good. It's been proven, proof of concept, ex vivo cells. You can modify them and give them back. We now, I think, have proof of concept. You can give some to go to the liver and modify cells there without necessarily getting a nasty immune reaction to the CRISPR protein, at least in some of the patients that I've seen reported. Whether how to use it in vivo into the brain or into muscle or nerves or something, I think is going to be more challenging. I think that's the only, we're going to run out of time here pretty quickly. We do have another 15, 20 minutes, but the other, and I know you got the next section is about antisense, et cetera. We want to get to that. The two questions in the Q&A right now are really about AAV. So maybe we go back to those, and I'm going to do it in reverse order because just I think that's worthwhile. One is I'm curious about treating multiple family members, i.e. with siblings with AAV, and they shed it. Will it make it impossible for other family members to get treated? And what do we propose as a solution to that?
Right. So yeah, the shedding. You know, in all the animal experiments I've done, I've never seen cross correction between siblings. You know, even if they drink the same milk from the same mom, and we're measuring the shedding in the clinical trials. Usually, of course, one thing that you could do is separate the treated family member, if at all possible, from the rest of the family for just a couple of weeks, because the shedding is really high only in the first weeks. You know, maybe don't let them share ice cream, and you know, certainly it's more prudent to keep the family members apart, but I don't think it would be necessary for an extended period of time. And the shedding can be measured with you know, just classic laboratory equipments. And then we would know most likely when it is safe to get back together. I think the bigger risk for AAVs is getting it naturally, because many of the currently used AAVs occur naturally. And so there is about, for example, for AAV9, there is maybe 15-20% of patients that have been exposed just by getting, you know, a viral infection. AAV travels with adenoviruses, enteroviruses, so sniffles, flus, stomach viruses. And so that is almost a bigger risk of getting positive for AAV. And then it depends on how positive you are, whether that would influence whether you can get the treatment.
Yeah, that's a great point. Probably impossible to avoid altogether, but certainly a major concern for families. And hopefully there are a lot of methods being used to or tried to get rid of those antibodies, or at least transiently reduce them and maybe enable dosing. So hopefully there will come a day, I'm confident we will have a day that we can overcome those. It's just what's going to be the best method and when will that day be here. The other AAV questions about delivery, maybe even a little bit more complicated. Can you comment on intravenous, intrathecal, intraperitoneal, or IP, ICV, ICM? I'm not even sure what all those are for CNS gene therapy. And also, do we prefer weight-based or brain-based brain mass?
Right.
Hard stuff.
Yes. It's complicated, right? So each of the AAV serotypes has different abilities to spread or not spread throughout the tissues. Some of the AAVs are also very good to be transported inside the cells. So for example, you can do an injection in a certain locus in the brain, and they can be transported through the cells to the cortex. So again, here I would say there's no general comparison. I think it really depends on the goal and what you need to target, the selection of your serotype. Generally, I'm a little bit more worried with the intraparenchymal. So intraparenchymal, you put the virus into the tissue directly. What I worry about there is that it can create local immune reactions, because you can only inject a small amount to not damage the tissue, and it has to be highly concentrated. So when you pull out the device where you put it in, there can be some leakage of the virus that goes back along those tracks. So those are some risks that you have with going into the tissue itself, or going even to the ventricles. You know, you have to go through the brain. And oftentimes you do that in patients that have a disease, and they already might have inflammation in the brain. So certainly that's something that needs to be considered. The intracisternal magno injections, which are here on the base of your skull, basically also go into the spinal fluid. So ICV, ICM, and lumbar intrathecal, they all access the spinal fluid and use the spinal fluid to spread the drug. And the ICM injections, I think, are potentially a good method, but they are complicated. There are specialized surgeons that can do those, and they are, I think, compared to the other two injection routes, maybe a little bit more dangerous because it's a small space to access, very close to the breathing centers of the nervous system, and if you damage something there, it could be detrimental. Also, there are certain patients that just anatomically you cannot do it, so it's a little bit more limiting. The easiest access point to the spinal fluid is certainly the lumbar intrathecal injection, which is basically what you do for any anesthesia that you give, women when they give birth or for other pain treatments in patients. And it's a standard procedure that every hospital in the world more or less can do. The problem is, of course, if you put it in the lumbar intrathecal space, you need to get it to the brain potentially, if that's where your disease is. And there are different ways to modify that. You can, the protocols in which I would say the spread of your drug in the spinal fluid is going to depend not only on where you put it, but it also depends on in what is it formulated in? Is it a heavy solution, very viscous, or is it a light solution that's lighter than the spinal fluid itself? Is it a large volume that you're injecting or a small volume? And are you injecting everything at once or very slow over time? So again, there, I think it makes sense in the beginning of a program to really think about the different options and maybe try a few things before you settle. If it is a drug that you want to make available worldwide, I think lumbar IT just has a lot of advantages from where I stand.
Tim, you're muted.
So sorry about that. There was a truck outside and I was trying to not have that interrupt. Yes, so this is one question is practical, and I'll answer this one as a clinician. How can we possibly think that we can explain such complexity in the informed consent process? It's got to be different than most clinical trials where there's a small molecule. And the only, this is probably something we could spend another hour discussing, and you have a whole panel of ethicists, et cetera. And I do think, yes, it is complex, but I will just comment that typically the informed consent is a process, not just at one meeting, not one lecture, not one explanation. There's a, talking to the family or the patients about from the beginning, they contact the site about the potential of enrolling in a clinical trial. So think of it as a process, multiple visits or touch points and ongoing explanations. But I'll also say, I'm not sure that it's possible. I'm not sure that any clinical trial we run really achieves true informed consent, because these are always complex issues, even small molecules and so forth. So I would say we do the best we can. And ultimately, there's some faith that the patient and family have to have in the process, in the system, in the scientific process, in the physician. So and in the committees that have reviewed the work up to that point, which there are often many. And maybe that's not the best answer, and the ethicist listening can perhaps create another session to discuss this. So I think it's a great point, but go ahead, Kat, we've got 14 minutes if you think you can zip through part of the next.
Yeah, it's short. It's very short.
Great.
I wasn't going to say much, but I thought it's worth talking about it because it's one of my favorite topics now. All right, exon skipping. Okay, before we go to exon skipping, just a real refresher. On the top, you have your genome. So your gene has sequences, which are those squares in blue, green, red, that will ultimately be part of the protein or will determine what's part of the protein. And then you have these large areas in between that we used to call trash, but we now know it's not trash. It contains a lot of information that is actually useful, and as of where is the protein made, how much, et cetera, et cetera. So from your DNA, from your gene, all of this is transcribed into RNA. But in order to make the protein, those sequences in between those squares are removed. And one of the big advantages is that you can actually make several proteins from the exact same RNA or from the exact same gene, right? So you can use all of the exons, 1, 2, 3, 4, 5, or you can use 1, 2, 4, 5, or 1, 2, 3, 5 here, and you get 3 different versions of the protein that can have different functions. So this is a tool how the cells can increase the diversity of proteins. That is very interesting. And we can use advantage of this also to use for therapeutics. So this is just a different representation of this, where those thin lines have just been kind of indicated by a little break here. So the introns in between. So the exons need to fit together in order to make sense. And this is indicated below with this sentence. If you put all of these exons together, you get a sentence that reads, "A big dog ran and sat." So that's when all of the exons are spliced into the mRNA, and then the protein is made, and it makes perfect sense. When you have a mutation and the exons don't fit together, the problem that occurs is that even though the first part is correct and the last part is correct, the overall protein doesn't make sense. And so suddenly it reads, "A big dog ran sat." Nobody knows what that means. And that's, for example, what happens in Duchenne muscular dystrophy patients. So if you could find a way to remove now a larger portion, so not only the orange exon like we had here, but also the blue exon, you're missing a bigger part of your protein in the end. But the sentence makes sense again. So now you can read, "A big dog sat." You didn't know that he'd run, but you know that it was sitting. And that's what we call a shorter truncated protein, which is partially functional. It might not be fully functional, but it is more functional than the completely messed up one that you just saw before. And exon skipping basically is the technology in which you try to achieve exactly that. You're trying to turn a situation like this, where there is a mutation that creates this mismatch, into a situation where you can restore this frame and restore the reading so that the protein does make sense. And it is shorter in that case. All right, so very quick, how do we do this? This is just showing you 3 exons here, and basically what you do is you block those sequences that indicate to the cells where they need to cut and splice together. And so you can skip one exon or several exons when you do that. And this is most often done with antisense oligonucleotides, but it can be done also with small RNAs, and it can also be done with AAV vectors if you code for the small RNAs that bind these areas here. So yeah, I think that's my last slide. So very quick about exon skipping. It's a method on the RNA level to remove pieces that can, so to make a shorter but functional protein.
And I guess we could add that that can be achieved by adding an oligonucleotide or delivering what is effectively an oligonucleotide with an AAV vector, for example, as you well know.
Correct. Yeah, very much so.
So it's 1:06. We have another 9 minutes we can chat. There's no open questions in the chat box. Would you like to make a summary statement of any sort?
Yeah, I think, I mean, the take-home message, I would say, is that, you know, gene therapy is a very diverse field that is also rapidly evolving, I would say, be it from the delivery standpoint and from the types of therapies that we can deliver. And I think it is exciting, but at the same time, I would say it is also, we have to be careful to overpromise in some cases. It is a very good tool, these are very good treatments, but unfortunately, in often cases, they are hyped up to be cures for diseases. And I think we're unfortunately just far away from it, because we would have to treat those patients probably at the time they're born, first of all, so that we can prevent some of the damage that will happen. And we are not going to be able to reach all the cells, right? So there's a lot of things to learn, but it is a rapidly evolving field. So I think on the hopeful side, the learning is happening fast from where I see.
Yeah. And, you did cover lipid nanoparticles that are lipids. There's a question about plasmids. What's the difference between a plasmid and a vector? And also, are ASOs more simple to construct than these complicated vectors? And maybe answer those real quick.
Right. Yeah. So a plasmid is basically a circular piece of DNA that is, discovered in bacteria back in the day, and bacteria can exchange those. They're almost basically chromosomes on its own. They can be pretty large, which is a big advantage, larger than what we can package, for example, in AAV gene therapies. And they can be injected, for example, naked directly into the cells with those shotguns or other methods. But they usually don't persist over time. So that is one thing. And again, the biodistribution is not going to be as good as something that's packaged into a virus. A nanoparticle is a particle, basically, that you could technically put an entire plasmid inside the particle. So you package it in order to better distribute it. So you can get it further with the packaging. And the packaging, the nanoparticles can be tailored to your organs of interest by modifying the surface, for example. So the nanoparticles you can put either proteins in it, RNAs, or DNAs of different lengths and shapes. So they are very flexible when it comes to that. Unfortunately, there's not as stable or as efficient as I would say for the delivery and distribution of treatments so far. But there's a lot of development that's going on in that area.
And I guess this seems like a relatively good last question to end on before turning it back over to Aidan. But is it fair to say, in our esteemed opinions, that at this stage of the game, genetic diseases can be so grave as to justify the significant risks they entail? And I'll let you answer it in a minute, but I would say our dictum still is first do no harm, as much as we can, the Hippocratic oath as a physician, as a scientist. And so the more that we can minimize the harms and the potential risks, then the more it's palatable to try to address that. It has always been the risk-benefit ratio. If it's a severe disease, then there are perhaps more risks that we're able to take. If it's an unmet need, if there aren't good alternatives, for example. And these are things that the IRB reviews and looks at, and particularly in pediatric IRBs, they rate the level of the risk of the study. Is it more than minimal risk, et cetera, in order to be able to justify and what's the potential benefit in order to be able to, both to the patient and to society at large for the research. So these are complex ethical questions and dilemmas that are debated every day around the country by IRBs and given significant thought. But I would say that there's been enough successes to justify, it's proof of concept that some of these do work. And so that helps, I think, in that equation, that there are real benefits to patients that can help justify that balance. What are your thoughts about that?
Yeah, I agree with you. I mean, I think, the more we use the technology, and that's why I personally I like to use viruses, vector serotypes, or flavors that are already in the clinic, because we've already seen what could potentially happen. It sometimes sounds attractive to use something new, but you have no idea what to expect. At least with the ones that we've tested a little bit more, the risk comes down because we do start to understand it better, and we do start to understand better how to deal with the side effects, or maybe, what is the upper level of what's tolerated. And there are, not only the approved therapies, but also several ones that are currently going through clinical trials that look really promising, that I think have profound effects, on the patients' and families' lives. So when you talk to the families that are affected by some of these really severe diseases, I think, yes, I think there is a real need and a real ability, hopefully, to make a difference.
I agree. And I look forward to learning even more in the future and applying our learnings to more and more patients as technology continues to improve and we can tilt that benefit higher and minimize that risk lower. Aidan, I think we'll turn it back to you.
All right. Thank you so much. And with that, we are out of time today. I want to thank our speakers for such an informative session and all of the attendees for your great questions. Please be sure to tune in for tomorrow's panel, Balancing Risks and Benefits, Gene Therapy Across Diverse Diseases, where we'll probably expand on a couple of topics that came up today. A registration link for that session has already been shared in the chat, as well as a link to view the rest of the sessions throughout the entire week of the Lunchtime Lecture Series. On behalf of PGTME, I want to thank all who have attended and participated or helped put on this webinar today. And I'd also like to remind you that PGTME exists courtesy of charitable donations. And if you're interested in supporting our educational initiatives, please contact our co-chair, whose name and email address will be shared in the chat. And with that, I will end the session and thank you all. Have a great rest of your day.